We will now discuss the second two levels of protein structure. Tertiary structure is the third structure, and that's when the chain starts reacting with itself or interacting with itself. For instance, this is one big protein. The primary is saying which each amino acid this is. Secondary is how it's being interacting with what's close to it. For instance, this is an alpha helix. Here's beta sheets. So those are small interactions. Tertiary is once it's got to the secondary, how it's interacting with itself and its environment. For instance, this is a hydrophobic region. That means that it does not like water. It's scared of water. It wants to get as far away from its water as it can. So it's kind of turning itself inward so that way it gets as far away from the solution as possible. Whereas this one is a hydrophilic reaction or hydrophilic interaction. Hydro, water again. Philic means it likes it. Notice it's turning itself outward so that way it can get closer to the water. We have another one right here. We have a carboxylic acid in this particular amino acid. And then all the way over here, if we follow the chain around, all the way around, it has a hydrogen amino acid there. That's the hydrogen bonds. Remember, a hydrogen bond is not a true bond. It's an attraction between electronegative elements and hydrogen. A salt bridge is here. Remember, an ionic compound is called generically a salt. And so here's a positive from the amine and a negative from the carboxylic acid. And it's interacting, forming a salt. So a salt bridge is bridging these two areas together because of the ionic interaction. Another one is right here. Those are disulfide bonds, meaning that the amino acid here must have had a sulfur in it. An amino acid here has a sulfur interaction, or sulfur in it. And so those two bond together. So there's 5 different interactions total that it can then start to ball up and form a shape. So primary tells you the sequence. Secondary tells you how it's interacting amongst itself. Tertiary is starting to form shapes. So here are the 5 interactions again. Cross links in tertiary structures. So it's linking itself within the structure. Hydrophobic is going to turn away from the water. It's going to get as far away from the water as it can. Hydrophilic is going to turn out and get closer to the solution. Salt bridges, you have the positive of one interacting with the negative of another across the way, forming a bridge between those two areas. Hydrogen bonds, a really polar element is interacting with a hydrogen along the other side, and it's forming an attraction there. Disulfide, if you have two groups that have sulfurs in them, they have a chance of forming a sulfur-sulfur bond. So, select the type of tertiary interaction. We're going to skip these because I did not make you memorize them. Globular proteins are spherical shapes. It's an example of a protein at the tertiary level. They do things like synthesis and transport. For instance, myoglobin right here transports oxygen. The hemi group right here helps to hold on to the oxygen. store and transport oxygen in the muscle. So this is an example of a globular protein. Quaternary structure is the final structure of proteins. Primary is the sequence of the amino acids. Secondary is how they're interacting with itself or with neighboring amino acids. Tertiary is where it starts to form a shape. Quaternary is multiple protein units. So it's multiple tertiary units that are linking together. For instance, with this one, this example is hemoglobin, and there are 4 peptide chains in there. So there's 4 tertiary compounds that are now being linked together, and those links are the exact same interactions as the tertiary. So if we go back up here, these 5 things, the hydrophilic, hydrophobic, salt bridges, hydrogen bonds, and disulfide bonds, are what's keeping these tertiary structures together to form the quaternary structures. So once again, here's the 4 layouts. Primary is a sequence. Secondary is within itself. Tertiary is when it is within itself, but in the more global scheme, bigger area. Quaternary is multiple protein units coming together. So here's the example again. Individual aminos, those are the primary. Then secondary is saying how it's going to interact with itself or neighboring ones. Tertiary is how it's interacting as a long chain, grouping together, starting to form shapes. Quaternary are those tertiaries being combined. So, beta pleated sheet, that's at the secondary level. Order of amino acids, that's at the primary level. Protein with 2 or more peptide chains, that's the quaternary. The shape of a globular protein, that's tertiary. Disulfide bonds between R groups, that could be tertiary, and it could also be quaternary, because remember, they said that it's the exact same interactions. They said just tertiary, but I'm going to argue that it's both tertiary and quaternary for the reason I just stated. Now we're going to talk about denaturation. We spent a lot of time talking about how the structures are put together. Now we're going to talk a little bit about how they're torn apart. We are going to specifically talk about these ways to break the proteins down. So denaturation is the disruption of the secondary, tertiary, and quaternary structures. So we're talking about we are breaking up not necessarily the peptide bonds, but we're breaking up the interactions between the peptide bonds or between the amino acids within the chain. You can heat it up or add an organic compound, and that's basically breaking the hydrogen bonds. You can add an acid or a base, which also breaks up the hydrogen bonds. Heavy metals tend to break up the disulfides. And you can whip it up, shake it up, agitate it somehow, and that stretches the chains out and the bonds eventually break. So, some more examples. An egg is cooked, that would be the heat's added. The skin is wiped with alcohol. You're doing an acid-base addition there. Heat is used. Instruments are sterilized in autoclaves. I'm going to point out the word autoclaves. That's actually adding heat to it. Autoclaves are actually used in medicine a lot too, in the medical industry, to sterilize medical equipment. So that's what they say, instruments are sterilized. So, for instance, scalpels or things along those lines, they'll put them in an autoclave, and it gets extremely hot with extremely high pressure to basically kill anything that's in there. So here are, once again, the 5 different ways a protein can be denatured. Heat above 50 degrees, so we're talking about adding heat, and this is how it's disrupted. Acids and bases, once again, you're interrupting the hydrogen bonds and the salt bridges. Organic compounds, hydrophobic interactions, meaning that instead of turning in, it wants to be turning out because it's going to be interacting with the organic compounds, because organic are nonpolar. Heavy ions, here are 3 heavy ions that it's referring to. Disulfide bonds are going to be broken up because of that. And then agitation, shaking it, whipping it, something like that. Hydrogen bonds, hydrophobic interactions, stretching polypeptides. So basically, you're just messing the whole thing up because you're pulling it apart. And that is the end of denaturation and the explanation of tertiary and quaternary.